Right now we'll proceed with some questions that I'll ask and pose of both of you just as discussion points that I'm sure will be taken up by the audience further in the future. And I'll just start with you, Dr. Reingold. What study designs do you actually think will be necessary to help test the hypothesis that gut microbes are contributing to cardiovascular disease pathogenesis? And do you think there are studies that may be unethical or versus ethical or logistically plausible to implement given the type of problem that we're facing with a disease that takes decades to develop?
So I'm not sure that I'm concerned so much about the ethics of these studies. I'm sure people have some ethical concerns about them. I do think there are much more cogent logistical problems. After all, as I think you pointed out to me in a discussion we had, it may well be that the important time when we're interested in the what's going on in the gut may be when you're a child. There may be many years of latency between the microbiome we're interested in and the cardiovascular disease outcome we're interested in. If that's the case, it's very difficult to see how you could do studies in humans that would look at that relationship. Obviously, for those who know epidemiology, we basically have experimental study designs where we randomly put people into different groups, and then typically we watch them for some period of time. Well, in this instance, the period of time could easily be decades or more, and those you definitely have to start those studies when you're young, if you intend to finish them, and they require enormous ongoing financial support. So leaving aside the ethical issue, ideally one would potentially randomize individuals at one stage or another, and then either measure their microbiome or try and alter their microbiome with some intervention, and then look subsequently, if not directly, at cardiovascular disease outcomes, what you think are important markers on the pathway or progression to cardiovascular disease outcome. I think perhaps more plausible might be what we call nested case control studies. But nested case control studies imply that someone has a cohort of individuals they're following over time, and that someone has cleverly stored away the relevant specimens in the right storage material, in the right volume that one can then go access at some point in the future. And I'm not sure necessarily that such cohort studies and specimen banks exist. But if they did, you could envision a study in which you took people with cardiovascular disease outcomes and without them, and then went into a stored samples and choose appropriate ones, again what we call a nested case control study, and look at materials that have been gathered in the past before the outcome occurred, and then see how they differ. I think you will undoubtedly have the huge problem of multiple comparisons. We've already heard about the fact that basically the statistical inference that we have is generally not well designed to deal with large numbers of comparisons, and the big data problem is going to be there, I think, unless we can refine the hypotheses much better than we have so far. That is, with much more specific questions about what's in the microbiome or which metabolic materials are important. Otherwise, we basically have an infinite number of comparisons to make.
Okay, Dr. Attie, what major obstacles do you foresee as being in the field to try and establish causality, as you were saying that's the thing that we're all trying to actually get at here?
Well, one of them is that the computational methods that are available don't scale very well. And so when you deal with as many variables as we're talking about, you very quickly become underpowered when you're dealing with large sample sizes, when you're dealing with large populations. So that's one issue. Another one is just an educational issue, in that this requires a lot of skills in computation and statistics and in biology. And there are a lot of people who have the computational skills and a lot of people who have the biology skills, but there are not very many people who straddle both sides. And I think for many of these very complex problems, we need to refashion our educational pipeline and give people a strong background in the biology and the computational side, because the inferences and the understanding is done in an iterative fashion, meaning that you do a calculation, you see the results, you think, then you turn on the side of the brain that thinks about biology, interpret that, pose a new question, do some more computation. So you go to the other side of the brain, back and forth. And it's very inefficient if it's being done by people who are overly specialized on one side or the other. So I think there's for all of what we call now systems biology, there's a big educational challenge.
Okay, before we open up for questions, I'll just suggest that why don't each of us indicate what you think your major take-home message should be on this topic. Dr. Reingold?
Well, I think a fundamental question is whether animal studies will suffice to answer these questions, or whether we need to do the studies in humans. And I know there's increasing skepticism about mouse models, for example. I know we don't necessarily have terribly good animal models for a lot of human disease. But for me, that's the fundamental issue. There's clearly been some very elegant work, as we've already seen, in terms of mouse models and fecal transplants and things like that. But I think a fundamental question is, would we be prepared to take data from animals and make inference about humans? Or are we going to insist on studies that take place in humans to really try and nail this question? Because if we really are going to require human studies, I think some of the complexities that we both alluded to already are really going to have to be overcome, and that the studies are potentially going to have to be quite large and take a very long time.
Dr. Attie?
So I'm not as worried about the animal model problem, because I think people are overly critical of results that don't extrapolate to humans, and part of the problem is that people are using inbred mouse strains, and so if you use one mouse strain, it's like studying one human being, and so of course one human being doesn't represent all human beings. So I think that I don't see that as a major issue. And the other thing is, if you break a problem down into rather than talk about a whole complex disease, you talk about the pathways leading to the disease, then animal models do represent humans extremely well. All the pathways are there. And I think that's how we need to break down these very, very complex problems. Can I ask you a question, Stan?
Sure, go ahead.
So there's I think a pretty good consensus that atherosclerosis is an inflammatory disease. And you've done some really elegant work showing that trimethylamine oxide is playing a role in that process. So the question is, given the great appreciation that inflammation is also involved in many other disease areas like cancer and autoimmunity and obviously diseases like arthritis, to what extent do you think this agent, the TMAO, is going to be playing a role in these other inflammatory diseases?
Okay, so I would argue that it's just the tip of the iceberg in terms of TMAO being one example of many potential contributory agents that gut microbiota are contributing to the pathology of cardiovascular disease. With respect to TMAO directly, there's some very intriguing data that has been coming out that looks at the connection between TMAO and cardiovascular risk in certain high-risk populations or cohorts. And I think that, so for example, we heard from David Relman mentioning that chronic kidney disease was a topic that wasn't discussed here. TMAO is excreted by the kidneys and the urine, and you see elevated levels in subjects with chronic kidney disease, and there's a manuscript that's in press that's actually due to come out. I don't actually have the embargo date. So, but that shows a mechanistic link between TMAO and both cardiovascular risk in chronic kidney disease, as well as the actual development of chronic kidney disease itself, in terms of the compound itself is linked to tubular interstitial fibrosis and impairment in function in the kidney with sustained high levels at the levels you see in CKD. So there are possibilities where it can have effects that are linked to alternative diseases, you know, like CKD is one where I think it's going to be particularly important. But I think there are many other microbial products that are going to be discovered, and it just takes, and that's my take-home point, we need to do the functional studies. Association only with sequencing and looking at associations, we're most of the time not even getting to the genus level in the microbiome studies. It's only at the getting only to the genus level, not to the species or the strain level. And I think many of the things we're talking about are not going to be a single pathogenic microorganism, but rather are going to be a conserved activity amongst a minority of microbes. So it's what are the biochemical pathways and what are the metabolites. With this, I think we can switch to the next section, which will be taking question and answer from the audience.